Hello and welcome to the 1.4 syllabus for this IB biology course. First of all, we'll talk about the types of transport and then we'll delve into the more specifics. So the first type of cellular transport that I'm going to talk about is passive. The most notable thing about passive transport is it does not require ATP to be able to occur. So we see in things like osmosis, in things like diffusion, both of which hold the same trend of a concentration gradient from high to low, which reflects on why there is no need for an energy source to move material in this way. The second cellular transport form is that of active, which of course is our involvement of ATP. Typically in cells, we see active transport through membrane-based proteins, so your carrier proteins, your channel proteins, which can also be involved in passive transport. A common example, which I'll talk about later, is the sodium-potassium pump as an example of active. Active transport is effectively the opposite in the sense that it goes from a low to high concentration gradient, which reflects on why ATP is required in this instance. So the first type of transport we're going to talk about is diffusion, which is your example of a passive transport. A definition of diffusion is the net movement of molecules across a membrane. A common example of diffusion in a real-world sense is, as an example, using something along the lines of an aerosol can. As you spray the aerosol can, the particles from the spray are diffused into the room, such that they are from a high to low concentration. So that's an example of diffusion. In a more cellular sense, diffusion is seen in things like respiration, with oxygen moving into a cell, or the opposite, with CO2 diffusing in the opposite direction, and we know that occurs with your alveoli in the lungs. So diffusion is this movement of substances across a membrane without the use of ATP, making it a passive. Another form of diffusion is this idea of facilitated diffusion. Facilitated diffusion is also passive, however, there is an involvement of proteins. An example of this is within the phospholipid bilayer membrane. Things like protein channels, looking something along the lines of this structure here, allow this facilitated diffusion to take place. It's imperative to note that it does not require energy. However, the protein may change shape as it comes in contact with its specific molecule. So facilitated diffusion is a very specific process. It cannot accommodate for an array of different molecules. Usually it's very specific, with an example being in cells, the use of facilitated diffusion for glucose. We know there are specific proteins within the cell membrane which allow this diffusion to take place in a passive sense. Things like GLUT transporters allows this movement of glucose into a cell, making it a facilitated diffusion and making it without that additional ATP energy source, which in turn allows more energy efficiency within cells. The next form of passive transportation is that of osmosis. Osmosis is defined by the movement of water across a semi-permeable membrane. Difference in solute concentrations on each side determine the movement of osmosis. We know that water moves with the solute concentration from a low to high concentration accordingly. There are different forms of osmosis. Firstly, hypertonic. Note the ER. This is where a solution with a greater solute concentration of another is found. So there is a higher amount of solute outside of the cell. than on the inside. And as we mentioned before, H2O always moves from this low concentration to high. So if there is a higher volume outside of the cell, then we can see here that H2O is going to move out, giving the cell, I've used an example of a red blood cell here, a more shriveled like look about it. And this is due to the water flowing out of it. Another form of osmosis is hypotonic. This is where the solution concentration outside of the cell is lower. Therefore, we can determine that water will be flowing into the cell because the solute concentration is higher inside. With this, we see an increase in cell volume. For hypertonic, we saw a decrease, this shriveled-like structure. So the increase in volume in turn creates a more turgid type structure. And we know this is the result of H2O flowing in. In other types of cells, we can look at things like the vacuole, which would in turn expand with more water, increasing the cell volume if the solution is hypertonic. Final form of osmosis is isotonic. Now, isotonic is unique because it says that there is an equal solute concentration in and out of the cell. This means that we're going to see an equal movement of water in and out of the cell. Now, an isotonic solution brings a constant cell volume, and this is because of this equal movement. So that is osmosis. As a brief summary for passive transport, we know that passive transport involves the use of small, nonpolar, and specific molecules. We talked about respiration and how passive transport involves oxygen and CO2. We talked about how passive transport can be used with small macromolecules, so things like glucose or other sugars. The role of osmosis and water, and how water flows with the solute concentration in that low to high. And we also talked about that facilitated diffusion with the use of integral channel proteins, so that thing like the GLUT transporter, which was mentioned, and how that aids this diffusion of such material. So I'm now going to talk about active transport, which we know requires this ATP. Active transport is a low to high concentration, which reflects on why it needs that energy. It allows specific internal conditions differing from the external environment. This use of energy means that the cell can be different from its external area through this low to high concentration medium of active transport. An example seen in the syllabus is the sodium-potassium pump, and its functionality is as follows. So, looking at the phospholipid bilayer here, we see that here is the presence of this pump. And the first step is the introduction of 3 sodium ions, which bind to the pump from the inside of the cell. So we can see that this is inside the cell, outside the cell. Here are our sodium ions. They bind to the pump. This triggers ATP phosphorylation. So I've used this red dot as our ATP. And effectively what ATP phosphorylation is, it is a phosphate molecule given to the pump from an ATP molecule, which allows the pump to have energy, which allows this active transport. So, after this ATP phosphorylation, the pump can change shape. On the second section of this page here, the pump changes shape such that it is facing in the extracellular direction. and this is as a consequence of that ATP. So looking down, we can see that change in shape, the potassium ion is bound to the pump from this extracellular medium. So there are 2 potassium ions, symbolized in green. Furthermore, we see the removal of these sodium ions from this pump. So effectively what happens is 2 potassium ions binds to the pump, and we know when it's in that extracellular direction, so we know the sodium is released, potassium is bound, and this releases the phosphate molecule. So as that potassium is bound, the phosphate molecule is released, and without that presence of the phosphate molecule, the pump is restored into this original shape. Following that, the potassium ion can be released, and then we're back into square one, where we can continue this cycle where needed. We're restored to the original shape, and we're ready for the cycle to undergo once more. A second form of active transport is that of endo or exocytosis. Both of these methods allow your larger molecules to move across the membrane. Both use the formation of vesicles to allow this to take place. As a result, both rely on this membrane fluidity for this fusion with the membrane. So we know vesicles are something which will fuse with the membrane, and then that's how they are sent out of the cell. And we know that the membrane fluidity relies on things like cholesterol, which is a good regulator for that, along with things like temperature and then the saturation of the fatty acid. So looking specifically at endocytosis to start off with, we know that this is bringing molecules into a cell. I've drawn a brief diagram to try to describe this, but effectively a portion of the plasma membrane, I'll allow this to be the plasma membrane, is pinched off. So we can see here, it's pinched off. It then sinks inwards and pinches inwards. So let the blue be a macromolecule of some sort. The membrane is pinched in, it sinks in, it's pinched inwards, and ultimately to the point where it's like this, where you have a vesicle containing your macromolecule inside the cell, and the cell plasma membrane is returned to its normal state. So looking at some specific applications for endocytosis. The first one is phagocytosis, which is your cellular eating. A very common example for this is white blood cells, and we know that they can ingest pathogenic material to then be ingested by the lysosomes. So this relies on this method of endocytosis to bring in that pathogen in the form of a vesicle, such that it can then be effectively transported into the lysosome. The second form is cellular drinking. And we know that some cells rely on this extracellular fluid, which can be engulfed by vesicles and then used accordingly. Potentially it might move into the vacuole or otherwise. Third method is receptor-mediated. And we know that receptor-mediated is typically very specific. We have things like receptor proteins, peripheral proteins these would be, on the external area of the plasma membrane in order to recognize these specific substances. Now, receptor-mediated endocytosis is used to acquire your more bulk quantities of substances. And I drew a quick diagram here to show how we have our receptors, which in turn allow this specific molecule to bind and then go through your method, which I described earlier, of this whole pinching inwards and ultimately ending up like that. So that's endocytosis. Now moving on to exocytosis. As a summary, it's effectively the reverse process. Now, exocytosis is typically used when something is produced inside the cell. So that would be, as an example, a protein made via ribosomes. So we know we have transcription of DNA creating mRNA, then translation at the ribosome. This is then sent and modified in the Golgi apparatus. So the Golgi apparatus effectively prepares the protein synthesized to leave the cell. It places it in that vesicle type structure and secretes it such that the vesicle can then fuse with the cell membrane, allowing your exocytosis to take place. So if we just look back up here, imagine the reverse process. You have your Golgi apparatus creating that vesicle, which allows that fusion, and it allows the steps to take place such that ultimately you have this on the outside of the cell. So, an example are things like hormones. Of course, there are other examples along the lines of just proteins in general, but hormones are a good example because of their application, especially so in humans. So, as a summary for active transport, we know it's seen in a few different ways, such as your endo and exocytosis. We know it's for your large and polar molecules. So unlike your passive transport, active transport is specifically focused on this type. We also know for things like ions, we talked about the sodium potassium pump. We know that active transport can be used there. Certain liquids as well. So things like cellular drinking with your endocytosis, that requires your active transport. And finally, of course, the larger macromolecules, which can't really be facilitated with your passive transport. They require an energy input. So certain sugars, certain proteins require that energy to be brought into the cell. One final thing I want to mention before the finishing of this video is osmolarity. We know osmolarity is the measurement of solute concentration, and we can distinguish the osmolarity of something based off its tonicity. So we know something with a high osmolarity is something that will gain water, and as talked about before, something that gains water is hypertonic. Low osmolarity will lose water, hypotonic, and equal osmolarity is effectively your isotonic with no water flow. So in terms of determining this on a more simplified scale, we can look at the volume of things. So if something gains volume, we know it's going to be hypertonic because it has more water. Vice versa for if it loses water, and if it stays the same, we know it's isotonic. So if we are given a real-world application or practical, we can determine the osmolarity based off the following.